So why don't I proceed? So today's conversation is to give you an update on some of the work we've been doing, some of what we shared with you historically over the years, but really specifically, about our goal to make it even easier to share your results and data from your genetic and genomic studies for complex diseases and whatnot. We make it easy already, I think, particularly for genetic data, because that's really our wheelhouse. But, with some of the things I'll talk about, it's been necessary for us to change that based on how we've worked with contributors over the past five to 7 years, and the growing change in our landscape in terms of the complexity and the requirements for data sharing. So we've adapted, and we think that the adaptation will really be helpful to you all as our users and contributors, but also to the resource to make it richer and more useful to our community that we share it with. So it's a sort of a two-sided win-win, and hopefully this will be useful. So today you'll hear two little presentations that give you some updates about that activity. But first, I'll give you a little context. And the context really sort of, to me, starts with the idea that, how is it that we built a resource where we have data from over 50 countries, representing close to 600 data sets for GWAS alone, spanning 600 plus phenotypes? This all comes from a partnership. And I can honestly say that for most of the data sets you see here, this was literally done with a collaborator, where we either emailed them, had many phone calls, long conversations, and direct contact with them, which is sort of the sweet spot of what we do best and what we enjoy doing the best. But getting the data in, getting it processed, making sure it's accessible in the right way, represented in the way the researcher intended, requires a whole software platform, right? And so the roots really started for us because way back in 2013, we were the data coordinating center for type 2 diabetes genetics. We were very lucky to do this. We were not only sequencing some of many of the samples, but we also were doing the analysis, and we were also responsible for making the data accessible to our collaborators. And this really formed the platform by which we figured out how to work with a collaborator to ingest their data, how they want it done, store it, protect it, keep it safe, and how to represent it. And this really sort of formed the basis of what we do, and also built the type 2 diabetes knowledge portal, right? So we worked with folks in these consortia to get the first knowledge portal built for type 2 diabetes with just 9 data sets. But for these, we had the individual level data, which was necessary at the time, right? So we built a whole machinery around analyzing that data and then representing it for ourselves, but also for the greater community. But what we learned, right, is that, over time with the diabetes knowledge portal, really just making the results and the summary statistics accessible to the world was really what the average researcher needed, right? The average person who's in pharma or who's in, a wet lab really just needs the human genetics results, not the raw bands and the sequencing files. That's too much for them. But you still need a place to store that. And in some cases, we store those things. But most of the time, it's just about representation of those results that are valuable to our users. So in order to do this, we built this sort of large-scale platform, which is not only a data ingest platform, but a software platform to represent those results. But the key feature here is that we work with, contributors, public data, you all, and our consortia that we're involved in to intake these data types, which I'll talk a little bit more, in 2 ways. We can take the individual level data, particularly for genetics data, right? Process it through what we call our loam stream pipeline, and then make it accessible through portals. But also, summary representations that I just mentioned are so much easier in many cases, easy to transfer, more important really to share with the broader universe. And that's another pipeline that we have. And we do that also in partnership with EBI. If there's genetic data that has to stay in Europe because we want to keep the individual level data safe in other geographic borders, but we can partner with EBI to make those results accessible and do the same analysis that we would do on premises here at the Broad and make those accessible to the world as well. The final piece is other data types, right? Not just genetic, but genomic, annotations, regulatory screens, and things like that. And this is where we partner with UC San Diego and Kyle Galton's team to build what we call the CMDGA, to warehouse, process, and store and represent all of the functional genomics data that is associated with these complex diseases. What's nice is that not only did we do this for diabetes, we formed a similar platform for other common metabolic disease areas and built other portals beyond that, also for musculoskeletal disease and type 1 diabetes and lung. So you were all familiar with this. This is something that's not new to any of you. But what's nice is the result of that effort, we are a registered NIH repository, which means that if you're writing your data management plans, which you're going to hear a little bit more about coming up soon, you can include us in them. And what's nice is we can partner with you to make sure all the results that you need to represent can be represented. And there's some pretty elegant ways by which we're thinking about doing that. One, from the registry standpoint, which Marie is going to talk about, and two, from the ability for you as a user to do it yourself if you wanted to, and build your own micro portals, if you will, which we call BYOR, or bring your own results. And we'll talk a little bit about that shortly, whether they are private to just your community you want to share them, or ultimately public, which you need to for many of these NIH requirements, right? But the world has changed over time since we did that the first time, right? The GWAS community has exploded with GWAS loci for, almost every trait, hundreds of thousands of these. And the GWAS catalog does an elegant job of making those results accessible. They do that better than anybody. Also, there have been technological and computational advancements that allow us to scale up the complexity of data that we can generate for other types of omics data, right? This is happening at scale all across our community. So what we've done, right, in many cases is we have a lot of ability to make these data accessible in our world. But what about this? What about all the data associated with getting to this point? This rests on this model where you have to know things about the regulatory element, the cell function, the organismal function, the cell states, perturbation, the screens, all the different variants. What are the causal variants? Lots of data go into this model, and that's complex, right? So it's a whole facet for each of the components of what it takes you to go from a variant, causal variant, to a disease. And what I'm saying by this is that as a result, we've sort of moved along with this field, right, to be able to represent more complex and more multifaceted data types. What else has happened? Well, the NIH has done something very smart, in my opinion, right? They've helped us. They've sort of moved the field to make all your results and all your data from all your studies more accessible. But to do that, you have to make them accessible to the world, but you need an easy way to do that, right? And in many cases, there are classic repositories. There's dbGaP, which you've heard about, right? There's ENCODE, which you've heard about. Some of them are super easy. Some of them are really hard, right? We want to work with you as an NIH repository. We're hoping to make this easier, but this is really hard, right? These are these long templates that you have to fill out. You have to submit them with your grant. Many of them haven't even been reviewed yet, so you're not even getting feedback because it's so recent. Moreover, then you have to execute on the plan. So, you must feel like this a little bit. It's host a new challenge and expectation as you're generating so much more data. So we're here to help. And we want to do it in the exact same way we've done all of our integration and working with partners to build resources before, which is hand in hand. Not making it a service that you just submit it to, and 2 weeks later you get an email back asking for correct this and correct that. No, we want to talk to you. We want to make it really easy for you to enter the system, share results, visualize them yourself, edit, preview, ask us for more, and then make them accessible. And that's really what we hope to do, both in helping you write your data management plan, but also in helping you execute it, which is really important. So broad classes, the data we support. Well, we support the data for all these activities in the resources we build, which start with the genetic associations, whether it's GWAS data, sequencing data of all types, credible sets, fine mapping results, that is, things from biomarkers or protein GWAS. So any of those type of biomarker or metabolite genetic associations we support. Functional genomic annotations, as I mentioned before, a lot of these large-scale perturbational screens are being generated. We warehouse those and make those accessible and visualize. You can visualize them as well. And also, anybody who does bioinformatic methods on any of these data needs a place to store those results. And again, often visualize them, make them accessible, make them downloadable through APIs and whatnot. So that's really sort of our main foundation of the things we can represent. We're lucky that we work with the Common Metabolic Disease Genome Atlas to store all of the genomic annotations, even the raw data and the process results, which is great for them. And what's wonderful about that is it's built on the ENCODE model. So if you do things through ENCODE and you want to store them here, you can do it in the exact same way that you've done before, and you can share between the two, which is lovely. So with that, we're going to talk a little bit about defining and executing your data management plan with the KPN. Broadly speaking, so Phoebe's going to talk about that next. She can talk about your data management plan, the template, some of the things we've done to give you sort of an FAQ if you're going to write one or execute. So where can you share the data? How do you go about sharing the various data types and results? So Phoebe's going to talk about that first, and then she'll turn it over to Maria, who's going to take you through sort of a more simple path to sharing your results, which is what we talked about before, which is the idea is you want to make them accessible in the way you envisioned. You don't want to go through this big knowledge portal process, which builds our flagship portals, but maybe you just want to, I don't know, visualize your EWAS results or your QTL results or your perturbational screen results for a portal that you want to publish as part of a paper. Well, this means can really get you to do this. This also fulfills the requirements of your data sharing plan, but it also allows you to have a portal that you helped create and you can make accessible to the world and iterate on as you go. So that's really the goal. We enabled these for you. They're free. They do require some collaboration, which we like, and hopefully that's useful because they're better for us, but you can also do them all by yourself. So with that, I'm going to turn it over to Phoebe and Maria. I also want to mention we have a new team member. It's always good to have a new team member. We have Farisa, who's pictured right here. She's going to join us for a year as a computational biologist to help actually bolster the sleep genetics portal with Richard-Sena. She has a background in that, but she's going to work on bringing all different data types. So I really want to welcome her. Maybe she's on the call, but you'll see her on future webinars talking about the sleep portal. So with that, I'm going to turn it over to Phoebe, who's going to walk you through data management plans. Any questions first before I dive in? We'll dive out, that is. All right, fabulous. All right, Phoebe, I will stop share and turn it over to you.
Excuse me. All right, thank you for the introduction, Noel. Excuse my voice. All right, so my name is Phoebe Alarin-Pamay. I'm a projects manager with the Portal team, and today I will be talking about how you, our users, can use the Knowledge Portal Network platform to develop and execute your DMS plan. So as mentioned earlier, the NIH rolled out a new policy on data management and sharing. The purpose of this new policy is to promote scientific data and to ensure that the fairness principles of data are being adhered to. So the NIH expects all investigators and institutions to plan and budget for management and sharing of your data, to submit a DMS plan when you apply for funding, and to comply with the approved DMS plan. The NIH also recommends the use of an NIH-approved repository, which we are, as Noel has mentioned. Now, this is very important to us because we specialize in the representation of summary statistics and end results, process data, and metadata for genomic and genetic studies. So if you are going to be applying for funding with the NIH, here's how we can help you to write a DMS plan. So before we get into that, I first want to talk about the elements of a DMS plan. First, you want to talk about your data types. What data types will you be sharing? Will you be preserving, representing, including metadata and also associated documentation? Second, you want to indicate what tools, software, or codes is needed to access or manipulate your data. You also want to describe what standards will be applied to your data. Then you need to talk about how your data will be preserved, accessed, and also provide a timeline. You also need to describe any factors that are associated with the access, distribution, and reuse of your data, such as informed consent. And finally, you want to describe how compliance with your DMS plan will be monitored or managed. So now you're ready to write your DMS plan. And what we've done is we've created an FAQ page that can help you with that. Here's how you can find it on the portal. So at hugeamp.org, under information, if you click on collaborate, you will find the DMS FAQ on the right-hand side, under data management and sharing. So we have created this page. It is new, and we definitely welcome feedback. We wanted to make it a resource since if you will be applying for funding, you will need to be writing a DMS plan. So we want to be able to help you with that. So just want to let you all know it's a work in progress. So we definitely welcome your feedback. Also, I want to know that this page follows the structure of a DMS plan. So we have included all the elements that you will be needing in writing your DMS plan. So what will you learn from the DMS FAQ page? You will learn about data sharing and upload into the CMDKP and the CMDGA. As Noel has mentioned, the CMDKP ingests and represents genetic data, summary statistics, but the CMDGA represents genomic data, including individual level data and metadata. You will also learn about data management and access, and also the regulatory compliance and data restrictions. So starting with data sharing and upload, we answer questions such as what kinds of data can I share and how do I upload my data set? We also let you know that while you're welcome to submit individual level data, only summary statistics will be made available. And then onto data management and access, we answer questions such as how will I protect my data? How do I ensure that my data will be made accessible? As far as regulatory compliance and data restrictions, for example, if you want to know what regulatory documents are needed to share individual level data, we have information about the IRB process as well as the data transfer agreements that must be executed. And finally, on the FAQ page, We have a sample of a DMS template that we created, which follows the elements of a DMS plan, so that you can use this to write your own. We're also able to assist you with writing your own. So we have our contact information right at the top of the page. You can contact us at help@kp4cd.org for any questions that you may have or for any assistance that you may need in writing one. So now Maria is going to talk to us about the data registry and BYOR and how you can represent your data and make it easy for you to openly share your genetic and genomic data.
Before we move on, that was great, Phoebe. Thank you. Any questions for Phoebe or comments? Because, you know, I bet you there's some of you on this call who have written them and may have some tips for us. Feel free to share feedback with us because, you know, we're learning this as we go. As Phoebe mentioned, we just wrote a couple for grants we've applied for. So we're kind of learning. So the one that's up there is one we've written for a recent grant. But, you know, I think this is a learning process, I imagine, and you know we're trying to focus it really on the type of data that we help represent, as Phoebe mentioned, but also you know think about as we talk about. Maria is going to talk about you know beyond genetic and genomic data, you know any types of you know biological results. We're starting to shift such that our software service, this BYOR platform, can accommodate that. So we're going to be expanding this as we go, and we really look forward to working with you all to make this useful and helpful as you you know meet with your compliance you know requirements from the NIH, but also you know do the fun work of being able to share the work that you do. So hopefully this will be a long conversation, a good one. Maria.
Okay, let me share my screen and start presentation. Okay, so first I'm going to say a few words about our new data registry that's in progress. It's not publicly available yet, but it will be fairly soon, I think. Okay, so oops, there we go. So I'm going to break this into two sections for genetic association data and genomic and functional genomic data. So say you are an investigator with genetic association data. Oops, I'm sorry, my mouse is very sensitive here. Okay, I won't touch the mouse. And you want to submit it to the Knowledge Portal Network. So the data registry will help you do that. Typically now, when someone wants to submit data to us, we have to figure out a way to accept the data. We can ask the Broad Institute to set up an Aspera transfer site, which is a little bit, you know, takes a couple days sometimes, and or else maybe the submitter has a Dropbox or something. But we don't have one very simple way to accept the data. And I just want to remind you that when you submit your summary statistics to the knowledge portals, we do a couple of things with them. So first of all, we integrate them into our databases. And that means that we meta-analyze them using our bottom line integrative analysis method. So we meta-analyze them with any other data sets that have associations for the same phenotype, and they go into the bottom line p-values. We also run a bunch of other methods across them. So then your data are in the knowledge portals. They'll both have an individual page describing the data set, and they will be integrated into the larger results that you see all through the portals. Now, an optional step when you submit your summary stats to us is that we can make a link to provide them for public download if you want. That is not an obligatory part of the process. And some authors choose to do it and some don't. You know, some want to have it ready when they submit their preprint. Some want to wait till the paper's published. Some never want to do it. So it's totally up to you. So the data registry is meant to facilitate this process. And I'm going to do a little demo of it in a minute. But there's another data submission process, of course, that many researchers want to submit their GWAS data to the GWAS catalog, and they should. This is a comprehensive repository meant to include all GWAS ever done. We would like to try to make this, well, sorry, the two resources are complementary, and yeah, we recommend that people submit their data to both resources, but maybe we can make this easier. So we're thinking about maybe we can connect our data registry with the GWAS catalog so that as you submit a data set, you could choose to submit it to the other resource or not. And I think this would be really convenient. We've talked with our GWAS catalog colleagues about it, and they like the idea too. It's not completely simple because currently the GWAS catalog requires a lot more metadata and harmonization before they will accept a data set than we do. Typically, we accept the data set in whatever format you have it with, with some basic requirements for content. The GWAS catalog is a little more picky, wanting you to validate the data and so forth before submitting it. So we'd have to bridge that divide somehow. But I think this would be a great thing for the future. And actually, yeah, if you think this is a good idea, maybe just drop a comment in the chat if this would be a feature that would be useful to you. So if you have genomic data, well, typically what you would do is submit it to the Common Metabolic Diseases Genome Atlas, our sister resource. And from there, we ingest certain the data types that we represent on the portals. We ingest them from there and represent them on the portals. And this is a list of all of the data types that CMDGA is currently accepting. So it's a lot. And so you can submit any of those to CMDGA, and then some of them will be immediately represented on the portals. Others may not be yet. So with the data registry, we are also hoping that it will be able to accept these kinds of data sets. But again, there's kind of a metadata disconnect, not a disconnect, but these data sets their formats are so varied and they have many different metadata requirements. So again, CMDGA has a process and a metadata spreadsheet to help researchers, upload all of the metadata that are needed for these data sets. So in this case, rather than duplicating that, what we're thinking about is kind of a two-step process where you would submit your data through our data registry, get an accession number, a link to download it if you want, and then from there it would go to CMDGA and the metadata and so forth would be collected. That's all still very in development at this point. So yes, if you have any thoughts about how you think this might work, how you would like it to work, we'd love to hear that. Okay, so those are my slides, and now I'm going to attempt to give a little demo of the data registry. This is our current prototype data registry. So I'll just go through this really quickly. So when you use the data registry, you would just give it a name. It doesn't really matter what it is. And you would choose whether this is open access, meaning that it's been published, or pre-publication. You give it a sort of more formal name. And again, these are names that we would curate in the knowledge portals anyway. So you don't have to think too hard about making the names consistent with the names we already use. I'm going to say test phenotype. Oh, there, I already practiced this. Then you choose your data type. And so we have all of the normal genetic association data types. We also have an other data type so that you can actually use this interface to upload, for example, a polygenic risk score file. And then if you choose other, you won't have to enter all these, the things that may not be relevant to a PGS file. Some of this metadata is not. So you would choose your genome build. You would choose an ancestry, sex, and then you can choose whether to, once you upload, get a URL that can be shared publicly. So for people can download your files, or to keep them private. You enter your sample size, and then you can enter a PubMed ID. I've got PubMed ID copied over here. Let's see. Okay, I think this is not currently working yet. Yeah, okay. What this will do in the future is it will fill out this description field with the reference, the title, authors, and so forth. So that'll be really nice. Have to put in your contact info and save. Oh, okay. Yeah, sorry about that. Okay, so as I said, this is a work in progress. Has to have some kind of description in there. Okay, and then you can upload your file. So here it will be one file per phenotype. And once you start typing, you'll get a list of our currently existing phenotypes. So if your phenotype matches something we already have, you can see the terminology we use to express that. And if it's dichotomous, then you can enter the number of cases and controls. Whoops. And finally, navigate to choose your file. And just upload it. And that's really all there is. When it's finished, you will get a link to it, and you can go back and edit the metadata if you want. So that this is was a really simple demo. And as I said, it's not, this interface is not finished yet, but we think it's going to make things easier to share data. Okay. Any questions about this part? Okay, let's move on then. I want to talk about how BYOR is getting easier to use. So as you know, BYOR is our service where you can create your own graphs and tables by uploading your own data. In fact, this variant sifter tool that you've all seen many times is created in BYOR. This is probably the most complex instance of BYOR that we have. But it just illustrates the point that it's got lots of different displays and tables. And I'm going to show you what's below the surface here. This is the editing interface for that page that I just showed you. And everything is formatted in JSON code. So there's this long string of statements. I don't even know exactly what you would call these, maybe statements or something, but if you're not an experienced programmer, it's pretty difficult to write this yourself. I mean, we have a tutorial that has templates and lots of explanations and stuff, but I've tried to do it myself. I'm not a programmer, and usually I end up leaving out one comma, which breaks everything. So the idea was to make an interface that would help people to do this. So I'm going to illustrate this also. So here, this is a list of predicted effector genes that I'm working on. And when you upload a table to BYOR, just a plain comma-separated values table, without any of that configuration, you can see the table. But you may want to do things like change the names of the columns, or put links in from gene names to gene pages in the portals, for example, or do some calculations on some of these values. So that all of that kind of thing is specified in the configuration. And that's when you have to write that JSON. So this configuration builder, which our engineer Annie Moriondo has written, is really it makes this easier. So you just paste in the first few rows of your CSV file that you've with your data, and you can step through and do all these different things very easily. So like here, these are the column headers of the file that I put in. And I can choose one and like rename it, give it a new name. And I can just step through and do different, I guess there's nothing in this table that needs a calculation, but I could set that up here and step through all the columns and do that. There's also an option to format the columns. So these are all the different things you can do, all the different operations that you can perform on what's in the columns. Then you can also set it up. I think you've all seen those nested tables that we have many of our effector gene lists in. So you can determine which columns are in the top row, and then which ones are in those expandable nested rows. Those are the feature rows as you go when you click the view button. You can also add tooltips for every column header here. And so as you do things here, the configuration is built here automatically. So all you have to do is just make all your selections in these user-friendly interfaces and then copy it. the configuration and paste it into the editor. So it's really simple. This will also write the code for filters, so you can add filters to filter your table by different criteria. So I think this is a really major step in making BYOR more useful for maybe the average scientist or the the non-engineer scientist. I certainly appreciate this. I've been using it a lot. So any questions about that? I think this is actually all I had planned.
Maria, can you kind of review for everyone? This is great, kind of how it all works together. Like, let's say they have data they want to both make a micro portal for and share it on the knowledge portal. Like, what would be the process from your perspective, just starting from scratch, just a very high level process?
Yeah, actually before that, I remembered that I did forget one thing, that DK is working on a page with many sections. So most of the BYOR pages you've seen, as you know, have just like one visualization and table. But DK is working on a method where you can make a really complicated page yourself with multiple visualizations, multiple tables. This is just a mock-up, but this will be really, really useful, I think, in the future. Okay, sorry, I forgot that. So, sorry, the process that people would.
Yeah, just to summarize the idea, like if ideally someone wanted to both submit data to build a micro portal, work with us to build that, but also ultimately wants to then make their data accessible in the knowledge portal fully integrated. They want to do all things, right? But they also, for example, are like, well, I have GWAS data and I want to visualize it with all those lovely functional annotations you've pulled together. Can you kind of walk them through what that experience would be like, just from a resource and a sort of process-based standpoint?
Yes, but DK, did you come on to answer that question?
Yeah,
It's fine.
I figured it might be too much for Maria, because there are so many technical things. So first of all, future plan for the data registry and BYOR. So first of all, the data registry, what Maria demoed is only for the part capturing metadata and then uploading the data files. But when you have smaller files, when it comes to the file size, there is no problem. You'll be able to upload in the files right away, and then you'll be able to access the files right away, right? Through BYOR. But what if you have a GWAS data or whatever data that is like huge, humongous, right? And also you need to you don't need to have like this million columns in your data to be displayed or used, or you want to just cherry pick some of the columns and build another files out of your raw files. That case, data registry will come with, we are targeting the end of this year, but it will come with data conversion functionality, which will allow you to pick the columns you want and then convert values in those columns and create new file out of the raw file. And then that will be still big. That case, you will be able to build mini bioindex, which is basically, let's say you have 100 megabyte file, then it's pretty hard to actually display that scale of data on a browser, viewer's browser. That case, we have a technology called bioindex. What basically does is it breaks that 100 megabyte file to very smaller pieces and index the path to the files by region or gene or whatever. And then you'll be able to query it through a pre-built API. So you can query the results and the data from those smaller files. So you can download the file in like half second, right? So the data registry will come with mini bio index and data conversion. And then when it comes to BYOR, so Maria demoed only the table building, table formatting process. But when it gets completed, you won't even see the infrastructure below the BYOR. You will be able to do everything on that GUI system. You will be able to create your BYOR page and then build whatever from there, and then you'll be able to link the pages you created through the configuration builder and combine them, actually link them, and build a mini portal out of it. So it will be much easier. I mean, I'm building a manual version of that multi-section BYOR page at the moment, and it doesn't have configuration builder for it yet. And then I'm realizing how painful it is. I mean, it was to do things manually. I mean, even though I built and I'm building the BYOR, I use configuration builder nowadays. I don't do it manually anymore. So it will be much easier and much more user friendly when things get done. I think that's it.
Great.
Yeah.
Great to hear it from the source, TK. Any other questions? I have a couple comments before people would log off and might be able to end early for people, but any other comments or anyone on the phone or the Zoom who's interested in being a guinea pig for us? We are more than willing to take you on as a participant. This is really something we enjoy doing because it helps us build other visualizations and learn where things go wrong or right in the process. Also, we are thinking of doing, specific boot camps and having tutorials. Mackenzie's going to work on a little video, kind of describe, what you can and can't do with it. So if you don't follow fully what we said today, because it was a lot that we threw at you, we have those available to you. What else? And we did mention that there's private portals as well that you can build. So if you want to make it private to other people, you can do that before you publish it. So I think that sort of summarizes where we are. I think, when is our next focus group or webinar, Maria or Mackenzie?
It will be a month from now. I will get that.
It's September 14th.
Wow. Well, back to the academic year, we'll be kicking out. Is it a focus group or a webinar, Mackenzie?
It's a focus group.
Okay, so we'll share with you a plan for that one as the date nears. And again, help@kp4cd.org is our email address. It comes to the faces that you saw on the call just now and many other people. And we get back to you. So if you have any questions, you can just pop those in that email and we're happy to help you. Even if you're like, I have some data, I have no idea if you can take it or not, reach out to us. We're happy to talk it through with you. So with that, I think we can end a little early today, unless there's any comments or questions. And we will give you guys back, 20 some odd minutes of your day. And we'll have this recording available on the websites, the various portals soon for you to look at. All right. Thank you, everyone. Thank you, Phoebe. Thank you, Maria.
Thank you.
Thank you.
Thanks, everyone.
All right.
Bye-bye.
Take care.
Bye.